Good morning, good afternoon, and good evening. I'd like to welcome you to this webinar, Painless Transition from Arrays to NGS and IDDD Analysis, introducing OGT's CytoSure Constitutional NGS solution. The presentations will be delivered by three of my colleagues at OGT, Paul Dougal, Sandra Catcher, and Jolyon Holdstock. My name is Dave Cook. I'm Senior Manager Regional Marketing at OGT, and I'll be your host for today's webinar. Before we begin the presentations, we just have a couple of housekeeping points to address. To type in a question at any time during the webinar, you can do this by clicking on the second button from the right at the bottom of your screen, the one with the three-dot icon. This opens up a window of options. Click on Q&A, and the Q&A window should open on the right-hand side of your screen where you can type in the questions. All attendees will be in listen-only mode to avoid any background noise during the presentations. So if you are experiencing any technical issues, then please type in the chat box. The chat box can be accessed by clicking on the third button from the right at the bottom of your screen with the speech bubble icon. This opens up the chat box on the right-hand side of your screen where you can send your message to the organisers. I'd like to introduce our first speaker, Paul Dougal. Paul has worked in clinical research settings in small biotechnology companies and is responsible for the Cytogenetics and rare disease portfolio of products at OGT. Over to you, Paul.
Thanks for the introduction, Dave. So as you mentioned, my name is Paul Dougal, and I am the product manager for the CytoSure range at OGT. Today I'd like to talk to you about CytoSure Constitutional NGS and its solution for IDDD analysis. Now I'm just going to stop my camera and move on to the slides. So, intellectual disability and developmental delay, IDDD, is normally discovered in childhood and affects people across the world. It has a prevalence of around 3%. IDDD is a catch-all term for a host of different constitutional disorders and disabilities with their own phenotypes and etiologies, where there's over 700 genes across the genome that have been implicated in these diseases, and the number is growing. The genetic heterogeneity and overlapping phenotypes can make absolute identification difficult. These disorders can be caused by large chromosomal deletions and duplications, like trisomy 21, as well as a host of medium and small copy number variants. Single nucleotide variants and indels have also been shown to be causative, and they often appear as de novo events. This graph shows the developing knowledge around the genes implicated in IDDD. You can see that over the last 20 years, there's been a dramatic increase in the number of genes being associated with IDDD. Now, this is likely caused by the improvement in technology and the level of information we gather from that. The genes are comprised of X-linked genes, autosomal dominant genes, and autosomal recessive genes. Research into IDDD tends to use a range of different tests depending on suspected disease type and information required for your study. If you're interested in CNV analysis, you can have single gene tests using technologies like FISH or PCR, where you use a single gene test to look at a particular known disease type. For instance, Fragile X or Rett syndrome. For improved CNV analysis, you have the chromosomal microarray, which is a higher throughput test and can analyze much high resolution for CNVs. So you can go down to the exon level for the CNVs, and also you can introduce LOH detection, so loss of heterozygosity with the array. There has been a trend now to move towards NGS platforms for constitutional analysis. And there's different ways to go about this NGS analysis. So you have your targeted panels, which are curated panels of genes that have been known to be involved in particular disease types. Then you have whole exome sequencing, which is where you detect the coding regions of the genome. Or a subset of that is exome panels, which are a slightly smaller subset. And then you have whole genome sequencing, which analyzes the entirety of the genome. And as you move down these tiers, you increase the information for SNV and indels, which increases the chance of a positive result. The question with the different approaches is the level of information you require for your study, and also how targeted you would like to be. Ethical issues arise using genome sequencing, whole genome sequencing, as you may have variants of unknown significance or benign hits, which require analytical time and declaration as well. So there's lots of different concepts to think about as you select your NGS platform. Here at OGT, we see an evolution of cytogenetics. So in the last 20, 30 years, we've moved from microscopic karyotyping, looking at G-banding of whole chromosomes, to FISH, where you have single gene resolution using fluorescent probes, and then onto the microarray, which has been the gold standard for the last 10 years. And this uses a range of different probes that can pull down CNVs at the exome level. Now, we see CytoSure NGS being the next step in this evolution on CNV analysis. Now, in terms of capabilities, you can split this roughly between two different avenues. You have your CNVs and loss of heterozygosity, and then you have your SNVs and indels. For the CNVs, the array has been the gold standard that we've mentioned before, as well as FISH. And then for SNVs and indels, you have exomes, so it would be that the whole exome or an exome panel, or targeted NGS panels as well. We see CytoSure NGS sitting between these two, as it gives you both that SNV and indel information you would expect to get from NGS platform, but also with CNV and LOH detection comparable to microarrays. So here at OGT, we have done extensive developmental work and corroboration studies to show that our CNV calling is completely comparable to our microarray calling. Now, how does one develop a panel? So we at OGT did this by looking at the literature, talking to academics, and really determining what genes are relevant to IDDD to be incorporated into our panel. So one influential study is the Deciphering Developmental Disorder study that was run, is still being run by a host of institutions in the UK that looked at over 30,000 samples of both parents and children to determine genes that are involved in IDDD. We also looked at ClinGen, which is a large database repository of known genes that can be involved in IDDD. And also OGT itself. We have over 10 years experience with constitutional diseases, and we've built up a large knowledge base within our company to really allow us to help curate our panels. So the key benefits of CytoSure NGS as a tool to investigate IDD samples is the fact that you can get that CNV, SNV, indel, LOH, and mosaicism analysis all in one tool, and all very much dedicated to looking at IDDD-specific genes. So everything you get from arrays and NGS in a single robust assay. We have that advanced panel design and the software to go with that. So a panel is only as good as an analytical software pipeline that you have. So we have an in-house software called Interpret, which is very powerful and has been designed to robustly and accurately call CNVs from NGS data. We have the most up-to-date content for ID and DD analysis. We've worked very hard, as I said before on the previous slide, to gain the most information about the genes responsible for IDDD, and we're the most current sample out there. This is a targeted panel of over 700 genes, which means you minimize that variance of uncertain significance over which means you don't get samples where you have to investigate potentially benign abnormalities or abnormalities that aren't known to affect IDDD, which saves on analyst time, saves on cost, and saves in storage as well. Also, we made this assay as cost-effective as possible, so you don't spend time and effort sequencing, storing. We include all the effort sequencing, all the effort for storing and analyzing data. We've made this very cost-efficient. So what is in this product? What do you get from CytoSure NGS? So you have a panel of hybridization capture baits for over 700 intellectual disease and developmental delays genes. That's 700 genes that are targeted for at the exon level, but as well as the 700 genes for IDDD, you have a backbone of genes, over 29,000 different regions that consist of the backbone, which gives you that C and V calling across the chromosome, similar to what you see in an array. You have the complete library preparation kit, which has been optimized and designed to work very well for this assay on the Illumina sequencer. You have that analytical software, which I mentioned before, Interpret, which comes complementary with the assay and has been developed extensively in-house to be able to do that C and V and LOH calling in complete comparative with the array. So you get that confidence that you have from OGT from our CytoSure Interpret software. This is built into an NGS platform. And this has all been designed to run on an Illumina NextSeq sequencer or higher. So this can also work on, say, a NovaSeq sequencer. We recommend around 24 samples for the NextSeq at high throughput, and that roughly provides, on average, 250x coverage. Now, just a little bit more about what goes into this panel. So we have almost 100,000 baits spread across the genome. 707 of these genes have been targeted at the exon level for that very robust calling of C and Vs in the IDDD significant genes. You have over 12,000 targeted regions. We include the UTRs in our bait design as well, so it goes to both ends. And then for the targeted region, we have almost 5 million bases targeted, and for the total panel, there's almost 9 million bases targeted. Now this slide here just shows you that distribution of baits across the genome. So you can see we have an even coverage. So from the larger chromosomes, so chromosomes 1 and 2, we have quite a high bait distribution. This then decreases as we go down the smaller chromosomes. So when you get to 21, 22, it's a lot less. And then for chromosome X, because of its influence in IDDD, there's a lot of baits dedicated to this chromosome. And it just shows you the thought that goes into the design of this panel. Now, I mentioned before about VUS and the issues behind this. So one of the key benefits of a targeted constitutional NGS panel is that you minimize this this unnecessary analysis time. So we focused very much on the content on genes and regions we know to be informative, which reduces those calls in the benign regions, and then it reduces that time taken to analyze these VUS, which, because you don't know what they are, you don't know the implications behind them, it can take a long time to review them. And then CytoSure NGS is approximately a quarter of the size of an exome. So you very much reduce the data size and analytical time dedicated to this panel. You save costs on both this data storage and, again, the analyst time. And we have, through the Interpret software we allow a lot of filtering and linking out to external databases with annotation, which means that you can really hone in on the criteria you have for your analysis. So on the right there, on the image on the right, that shows you some of the filtering and protocol settings we have, which you can really kind of dedicate and optimize to your analytical pipeline. And then on the left, we have some of the filtering options that we allow, which then reduces those hits you get on your analysis. Now I just want to give you an example of how this data looks and the effort's gone into the kind of user design. So here we have a 2.1 MB deletion on chromosome 1, and you can see it looks very much like an array software presentation. So you have LOH calling at the top, and then you have your CNV calling on the bottom there. And you can see this has been designed to look a lot like our CytoSure Interpret software, which shows you the deletion by the drop in the ratio plot there. So you got a clear deletion sample, and then the LOH, we can see that separating from the allele, the allele plot. So we've made this as easy to use for the cytogeneticists as possible. We've made it look as comfortable as possible, so you can easily transition from array software to this NGS software and being able to analyze it as quick as possible, and very familiar as well. Now, this is an example of how the S&V data will look. So we have the alignments on the bottom there of the gray bars. And this example is an insertion with the JAG1 gene. It's just a small duplication. And you can see how the software presents this. A nice table format at the top. And then you have the very easy to view IGV viewer at the bottom. Now, we ran extensive trials on this software. We've run a lot of corroboration studies to make sure that the CNV calling is robust as possible and is directly comparable to the arrays and their software. And this is just some of the recommendations we've had from people who have tried our assay. So Chus in Canada is a hospital there, and they are very happy with how the CNV data looks. They are happy with the resolution you can go to, so we can go to deletions smaller than 500 BP, so you get that extensive resolution at the exome level, and it's easy to run as well. So our assay is designed to be as easy to run as possible, and my colleague Sandra will talk about that later. So again, they said that the support and training as well is outstanding from OGT. So OGT, we're there for the customer. We want to partner with you. We want to help you with your analysis needs. And then EGL, Emory and Genetics in the US. Again, impressed by how easy the assay is run. And then it's basically just the quality information that comes out of there as well. So that's me. So if I just pass back to Dave.
Many thanks, Paul. I'd now like to introduce our next speaker, Sandra Catcher. Sandra is a senior scientist in the development team, having worked for 12 years at OGT. She was involved in protocol development for CytoSure Constitutional NGS and supported the training of beta trialists at customer sites. Over to you, Sandra.
Thank you, Dave. So I'm just going to spend a few minutes outlining our poster presented at ESHG in June this year. I'll be taking you through some of the wet lab processing of our CytoSure Constitutional NGS panel, and I will then show you some of the results generated in our R&D and alpha and beta trials. I'll just move over to my presentation now. So here is our ESHG poster that we presented earlier this year. This is our instructions for use protocol, and you can find it available to download on our website at ogt.com. Here at OGT, as Paul said, we do offer you the complete solution. We provide the necessary kits for the library preparation, hybridization, wash, and all the bead purification steps, which includes the Ampure XP and the streptavidin beads. We also have our analysis software, Interpret. The kits come in two sizes, the 24 reaction kit and the 96 reaction kit, which also includes enough excess to still run your samples in batches of 24. So our protocol starts with 24 1 microgram good quality genomic DNA. This is then sheared mechanically with a Cavaris machine. The library preparation then follows with end repair, A-tailing and ligation of adapters. Our new improved workflow now includes an indexing PCR prior to hybridization. At this point, it is important to make sure that your indexes are unique within your sequencing run And for ease of use, it is possible to multi-channel a column of indexes per 8 samples from our indexing plate, ensuring that less mistakes are made. The 24 samples are then pulled into 8 prior to hybridization, dried down and then hybridized overnight, leaving you with 3 tubes to be captured, washed, PCR'd and ready for sequencing. Our recommended sequencer is the Illumina NextSeq, and a high output kit will provide you with the recommended coverage of 250x across 24 samples. Our panel has also been used on the NovaSeq, which you can sequence up to 48 samples in one SP flow cell. This pre-pooling method greatly reduces the complexity of the lab work and thus saving hands-on time. The complete workflow takes around 3 lab days, from fragmented genomic DNA to the generation of FASTQ files. It only requires 8 hours of actual hands-on time. So let's take a look at our results. We studied a total of 214 samples, 72 of which were commercially purchased and contained known positive and negative samples. The other 142 samples were research samples donated from genetic laboratories with known aberrations and were either processed in-house as part of our alpha trials or at the customer site as beta trialists. These aberrations were previously identified by microarrays, as in the case for CMVs and LOH, or sequencing for SNV and indels. So one of the advantages of our product is that you can identify high resolution events like SNVs, indels, as well as larger aberrations such as CMVs and LOH. My colleague Julian will take you through our software in more detail, but here is an example of a 4-base pair deletion identified on chromosome 16 within the anchoring repeat domain 11 gene. And here is an SNV in the GJB2 gene on chromosome 13, and a very obvious SNV in the IGV panel below. Here is a 66 kb duplication for chromosome 2, and on the bottom you can also see the array confirmation. Our software also identifies large aberrations such as this duplication event and an LOH on chromosome 7 in this Coriell sample, NA3098. Here is an example of a large deletion on chromosome 1, and inversely a small 150 base intragenic deletion on chromosome X. A small number of mosaic samples with CMVs were also included, and the detection of these is dependent on the size of the aberration and the level of mosaicism. A criteria for including mosaic calls was that the aberration had to be greater than 5 megabase in size and a mosaicism of 50% or more. Here on the left is a mosaic deletion of 7 megabases on chromosome 15. If the aberration is of a large size, as in the aneuploidy plot on the right, which shows a 10% trisomy 9, whilst it was not able to be called by our protocol as a mosaicism was under 50%, we are still able to visualize the aberration manually and quite clearly by summarizing the counts over the whole chromosome. So here is a summary of our findings. This table illustrates the results obtained in the study, with the number of samples where the pathogenic aberration was called by the software and the percent sensitivity for each type. As you can see, CytoSure Constitutional NGS solution is able to detect pathogenic CMVs and LOH with a performance on par with microarrays, as I have demonstrated, whether it be large CMVs or small intragenic CMVs. We also called 100% of reported pathogenic SNV indels, showing the precision and sensitivity of the assay. CytoSure Constitutional NGS's ability to call both SNV and CMVs gives this assay a larger scope in calling genetic aberrations than in just microarrays alone. And that's it from me. I'll now pass on to Julian, who will give you a more in-depth insight into our Interpret software. Thank you very much for listening. Back to you, Dave.
Many thanks, Sandra. I'd now like to introduce our final speaker, Julian Holdstock. Julian has 16 years experience at OGT as a software developer and data analyst. He runs the team that developed the Interpret software to support analysis of OGT's targeted NGS panels. Over to you, Julian.
Thank you, Dave. As Sandra said, I will give an overview of the software and have a bit of a demonstration at the end to show you some of its capabilities. So I'll just switch to my slides now. Okay, so as we said, OGT has extensive experience in providing individualized options through its established plugin infrastructure. This enables software to be tailored to your laboratory's specific requirements. And this is one of the features that we have with the Interpret software. Additionally, there's a comprehensive range of dynamic filtering options which are available, allowing you to be either very broad or very specific with the searching and filtering that you want to implement within the software. And at the back end, we have a relational database which will store all the activities conducted by the software, so all results and variant calls are retained within the database. This enables logging and tracking of individual user activity for consistent data processing and laboratory monitoring. And we can also use the database to implement security protocols such as access permissions to different parts of the software. And so used in conjunction with our SureSeq and CytoSure NGS panels, Interpret is able to facilitate analysis and visualization of a wide range of mutation types and structural variants. So this complements the expert panel design and hybridization-based approach that we've developed in the lab. So as an overview of the process, you have FASTQ files which will be loaded, and once within the software they can be processed by the pipeline. The pipeline itself is very flexible, it can be configured along certain parameters. Additionally, you can specify particular metrics, so you can decide what you will feel is an acceptable quality of data that is generated, and you can also include filters in the pipeline to remove or target specific variants of interest. And once completed, this data is stored in the database, allowing flexible reporting of the information that has been generated. For example, you could generate a summary report as an overview of the batch. Alternatively, you could have a QC report for focusing on the quality of the data, or you could have a full report with all the information that's been generated. And we have the ability to generate different types of report according to individual requirements. So if we look at the software itself, here's an example of the output of a batch. And you can see as part of the results, there is a batch QC report generated. And if you select that, it will open a separate window with a multi-QC report. You can see the different types of variants that have been detected. So here we're reporting SNVs, CNVs, LOH, and translocation events, and you get to see the different numbers of events in each sample that have been generated. Besides the batch QC, batch report, you can also have an individual sample QC report as well if you want to drill down into an individual sample. And then lastly, this view is highly configurable. Wherever in the software you see the three horizontal lines within a button, that provides the opportunity to edit the columns which are displayed. And I can demonstrate that when we switch to the live software in a minute. And then if we look at an individual sample, you can see we have a variant view, and the software view is divided into different sections. We have the protocol filter. So this is the filter that is implemented as part of the analysis pipeline. You can see on the left-hand side, we have the number of variants that go in, which is 2,757. And on the right-hand side, the number of variants that come out in the green. As you can see, the default filter here is all variants, so nothing is going to be filtered out. And then below that, we have a variant table. So this will show all the variants that have been detected, 1 variant per row. And if you select on a variant, you can see that the information and the alignment is loaded into the IGV browser window below. As Paul said, we're keen to work with customers and to improve their ability to work with the software and to find what they're looking for. And one of the early bits of feedback we had was to, instead of having a sample-centric view, to have a variant-centric view. And so this is something which was recently been implemented. So it means that instead of looking at all the variants in one particular sample, you could take a particular variant and look at how many times that has appeared in different samples. So as with the previous view, this is divided into sections. There's another filtering part. This is actually a dynamic filter because now we've moved beyond the pipeline. This is a filter where you can filter the variants which have been found and are stored in the database. Below that, you can see here is the list of different variants. You have the first column showing the HGVS annotation for the variant, and then the next column you can see the number of samples that have in which this particular variant has been detected. If you select on a particular variant, you can then see all the samples which have been found to contain that variant, and then below that you can select one sample or multiple samples to see in the IGV window below. And now I'll just switch over to the software itself so we can have a slightly better look at it. Once you've logged into the software, this is the dashboard you'll come to. We have a series of menu items along the top, but we also have some shortcut buttons along the bottom. The software, to run it, is relatively simple. You want to upload your FASTQs, you want to run a batch, and then you want to view the results. So to upload samples, you can either use the file browser. Alternatively, you can just select individual FASTQ files, drag them in, and these will automatically pair up based on the Illumina nomenclature. You can check that you're happy that the pairs have been assigned correctly, and then you can upload the FASTQ files into the software. Once samples have been uploaded, then you can start the analysis process. In order to start the analysis, you need, besides the samples, you need 3 things. You need to specify a batch name. Here you can see that it defaults to providing a batch name based on the date and time of the analysis being run, but an alternative, you can type a different name in if you want to. Secondly, you need to choose the relevant panel that is being analyzed. So here we can choose CytoSure NGS panel, and then you need to choose the protocol. The protocol is the settings that are used by the pipeline and also includes the metric set and then also includes things like the filtering options. And I can go into the individual protocol a little bit later on. If we choose, say, for example, the default protocol, and then you can add samples to the analysis, and you can see as you add samples, they're added and shown in the table below. And once you're happy that you've got the necessary samples and you're running the correct panel and the correct protocol, you can click Run Analysis, and that will start running in the background. So if we look at a run that's finished, you can see that this will be like the slide I showed earlier. This is the batch output. You can see we have an overview window showing who ran it, what panel was run, when it was run, and its status. You can also look at the protocol that was used. So it enables you to track everything and how it was done. And you can also look at the multi-QC report. So if you select that, you can see you'll get an overview of all the different samples that have been processed in that particular batch. Additionally, you can look at file status. The software will generate a large number of files, starting with the alignment file, for example, the BAM file and the BAM index file. It also generates QC files, VCF files, and additional CGH files, which is a feature requested by our previously existing microarray customers for the microarray software. We also have output for translocations and then a log file. So this means that if you wanted to do something with the files that have been generated, you can download them individually or in bulk and use them for an alternative process if necessary. You can also generate a report. We have different types of reports. So, for example, you can generate a batch report, or you could generate a PDF report. And then once you've selected your type, you can choose a template. These are very configurable. And what we found with other softwares we generated is individual customers will want individual types of report, but we have a lot of experience generating the different templates. And so we'd be able to work with you, find out what you needed, provide either the report type or the template, which you could then upload and then use. So for example, if we then generate the report, you can see here, this is just a very basic report showing a PDF of the output of the results for each of the samples and showing specific run metrics, but this is very configurable. And then you can see we have the samples themselves. We have, for each row, we have an individual sample. You can see you can select to view the SNVs or the CNV events. We have the different numbers for each of the events which have been detected. You can generate an individual report for that particular sample or a QC report. And as I mentioned before, we have this little 3 horizontal barred button here. And if you select that, you can add additional columns to the software to look at, to generate additional data. So if I select a sample and we can view SNVs, you see along the top, it's the variance page. You can either return to the batch or you can switch to viewing SNVs or translocations. So you can move between the different types of variants for each sample. As before, you can see we had the protocol filter. And the protocol filter is showing all variants, so nothing is filtered out at this stage. We have the row below, or rather the table below, showing all the different variants. And if we select a particular variant, you can see that it is loaded into the RGB viewer below, and you can see the actual alignment. And so this gives the opportunity to kind of have a better look at the type of data that has been generated. Within the variance table, we have different functions. So you could generate a report, you could generate a shortlist, you can filter. Alternatively, you can also manage tracks. So you can see in the RGB viewer below, we have genes, which is a track which has been loaded into the software. If you have other tracks of interest, these can also be added and displayed in the RGB viewer as required. So you can also modify the columns which are displayed. You can see here we have another of these 3 horizontal barred buttons. So you have a wide range of different columns you can choose to view or not to view. You can also click on the variant itself to add it to a shortlist. Alternatively, you can set a preferred transcript. As a rule, the software will report on the largest canonical transcript for a gene, but if you have a particular transcript of interest, then that is something you can set. You can also classify. For example, you might decide that it's benign, in which case it will become colored in the color denoting benign, and this variant, if it's displayed in any other samples, will also be highlighted with the same classification that's been given. And classification is something that can be modified. One of the other features which we touched on earlier is the ability to filter. So if I go back to the action and select the filter option, this accesses a large number of variant attributes that can be selected in order to filter down and drill down into your data. We have basic variant attributes such as sample, chromosome, start, end, etc. There are variant call attributes, and then we also have consequence, severity, population frequencies, classification pathogenicity, genes, exons, and proteins, or there could be region lists or variant lists that are of particular interest to you that you can specify and filter on. So, for example, we look at just selecting a set of chromosomes, so you could just be interested in say chromosomes 1 to 5, and then you can add that as a filter. Besides using these options here, there are also the opportunity to use previously found options. So you might want to, for example, add in something which has been defined before, and when you do this, you can either add it as an intersection, so essentially both criteria need to be satisfied, or you could add it as a union, in which case it would be or. So you can, in this case, you can say it's either chromosome 1 to 5 or pathogenic, and you can update the bottom. And you can see now that before, where we had our protocol filter with all variants, we've now got a dynamic filter which has been added, which is filtering based on these specifications. And you can see that we've gone from 3,500 variants down to 1,075. And this is a basic example of what can be done, but there are very sophisticated and complex filters that can be built up and deployed as part of the process. That's probably all I wanted to touch on. Very quick overview. I would normally spend more time, and I'd be happy to arrange a time to go through the software in more detail individually and have a real deep dive. But in the meantime, I'll hand back to Dave.
Many thanks, Jolyon. With that, we have been monitoring the Q&A box for any questions for our speakers. Okay, we're just having a look now. First question, can this kit detect point mutations? I guess that's a question for you, Paul.
Thanks, Dave. So the simple answer is yes. So this is an NGS-based kit, so it's able to detect those S and Vs, so the single nucleotide variants, as well as insertions, deletions, and then we can assign them different types of mutations, so we can call them frameshift mutations or missense or what have you. So yes, we definitely have the capability there.
Okay. Can the CNV calling actually be as good as an array? Paul.
So can you repeat that, David? I missed that one.
Yeah, sorry, I've got some echo there. Can the CNV calling actually be as good as an array?
Yes, so we have performed extensive testing on the accuracy of our CNV calling, and we've conducted tests in both in-house and externally in independent labs across the world, and we compare our NGS data with their array data using the same research samples. And we can see, and Sandra touched upon this on the aCGH poster there, we've got some excellent corroboration with the data, and we're very confident in the robustness of the CNV calling.
Okay. Sandra, we don't have a Cavaris. Is there an alternative?
Yes, absolutely. We are currently working on an enzymatic alternative, and we are hoping to make this available in the near future. So, in the meantime, before it becomes readily available, feel free to talk to us and we can help you work on your protocol together.
Okay, thanks, Sandra. Jolyon, how can I create a filter for a sample and use it on a protocol?
What you'd do is you'd start with a basic protocol which let all variants through. You could then play with the filtering options you have and develop the filter of choice. once you're happy that this filter is able to suitably reduce the number of variants that are called by the pipeline down to the sort of more manageable number or the ones that you're specifically interested in, you can then save it. And that means that if you wanted to create your own protocol, when you create that protocol, you could specify the filter you've created rather than the one which lets all variants through, which is the default setting before you make any modifications. And once you've got your filter in place, then it'll automatically use that in all future runs.
Okay, thanks, Jolyon. Paul, our lab doesn't run NGS. How easy is it to use?
That's a good question, Dave. Thanks. So we've designed this platform to be as user-friendly as possible. So that includes the design of the reagents and the kits itself. We've got excellent feedback on how we label them and how it's set out, so it makes it very simple for the technician to use. And then the methodology of the assay as well has been optimized as much as possible, and that's an ongoing process to increase this. And then the interpret software as well has been designed, and I mean, we showed this earlier in the presentation, Jolyon and myself. It's been designed to be as user-friendly as possible and as familiar to the cytogeneticist as possible. And then alongside these technical steps, we also provide hands-on training with our excellent application scientists who will be able to train you and troubleshoot any issues that you may have. I think that OGT really believes in partnering with labs to get the best information possible from their samples.
Okay, thanks, Paul. One for Sandra here. Are there any suitable stopping points in the protocol?
Yeah, that's a very good question. Yes, absolutely. After every process QC that we do, that is a stopping point. So you can split the protocol into much smaller chunks if you wish, and do it over a couple of weeks or whatever you need to fit in with your lab's requirements.
Okay. Someone's asked about, I presume with you, Sandra, where can I find a list of genes and targets for the panel?
We don't publish that on our website or anywhere, but I can take down your details. We can see who's asked the question. So a member of our team will be in touch shortly and we can email you through a list.
Okay. Thank you. We've got one, Jolyon, from looks like they're an existing CytoSure user. Can they use their CytoSure database with the Constitutional NGS?
Yes, we can, or rather, yes, they can. Because we designed that database, we know its structure, and there is functionality within the advanced settings in the software that allow the software to connect with that existing CytoSure database and use the data that's already been generated over however many years previously. It's not to say other databases can't be connected, but that would be more of a kind of bespoke bit of coding, because we find individual customers' databases are as the customers are individual, and so we would need to tailor a bit of code. But that's certainly not impossible to do in other cases such as that.
Okay. Let's just. Yeah, I presume this is for you, Jolyon, as well. What hardware specs do we recommend, and what operating system can it be used on?
Currently, the software can run either on Windows or Unix. From a sort of spec basis, we'd say at least 16 gigs of RAM and 8 CPU cores. But I would say anything more than that is a bonus. The more resource that's available to the software, because it's multi-threaded, the quicker that analysis can run. And from a data storage point of view, we'd say at least 1 terabyte, but you have to, I think, bear in mind that the more you use it, the more data you will generate, and the more storage requirements there will be. So I would say as much data, as much space as you can find, but start with 1 to 2 terabytes, and then have the ability to expand it later if possible.
Okay, thanks, Jolyon. Paul, one here for you. Do you only detect CNVs on the targeted genes?
Okay. So while this is a targeted panel designed for IDDD analysis, to have the optimal CNV calling, we have almost 30,000 SNPs across the backbone. And that's similar to what you see in the array where you have the backbone spread to get that better CNV calling for the larger CNVs. So not only do we have the 700 IDDD genes at the exon level, we also have this larger range of genes at the gene level, which allows us to call and pick up larger CNVs across the genome, but just a slightly lessened resolution than the targeted genes. So yes, you can definitely pick up other regions with this kit.
Okay. Here's a question, Paul. I expected to actually sort of see at some stage. Yeah, can you compare the cost per sample for array and for this NGS solution?
Yeah. So this is one of those things. It depends. It all depends. So there's lots of different things that go into these kits and the parameters. We have made the reagents as comparable to arrays as possible. So it's not going to be a million miles away from your current array spending. But then you have the sequencing costs on top. But if you look at basically the cost of a good kind of research result or a good sample result, it becomes much more cost effective because we have that extra information from the SNV and indel analysis as well. So you have that greater depth of information, which means you can call more things without reflexing to other tests. So in terms of cost per sample, it becomes very reasonable, or reducing cost per sample for the arrays in terms of positive results.
Okay. Okay, thanks, Paul. There's another one here, I presume for you, Paul. The list of genes is constantly increasing. How can we stay up to date? Can genes be added or changed at some stage?
So currently we have a set gene list based on, as we talked about in the presentation, we looked at different places and we curated the best gene list possible for the current information. But you're right, as more genes enter into the system, we'll have to update. But we don't predict to be doing many updates currently, so it'll probably be very few in the next couple of years because people don't like to constantly be validating their processes. So we will update if there's a great reason to, but currently there's not room to add additional genes currently.
Okay, right. Thanks very much indeed. We're just looking at all these questions. Yeah, another one, I presume for you, Paul. Do you sequence all the exons?
So we sequence all the exons on the targeted genes. So that's around, I think that's around 12,000, just over 12,000 exons we target over the 707 genes. So yeah, we target all the exons for the target sections.
Okay. Question for you, Jolyon. And how much actually does the software cost?
That's an easy one. The software is free. As long as you are using OGT panels, then the software is absolutely free.
Actually,
and as is the support for it too.
Okay. Can you actually use it with other manufacturers' panels?
Currently, no. At the moment, the bed file that will define where the software will look for variants, that format is encrypted, and so it needs to be provided by OGT. and currently we only provide the files for our own panels.
Okay, thanks very much indeed, Jolyon. Well, we're coming towards the top of the hour now, and so I feel we've reached the end of our webinar. I'd like to thank our speakers, Paul, Sandra and Jolyon, and to all of you for joining this webinar today. If we didn't have time to get to your question, we'll look to get an answer for you after the webinar. And finally, if you missed any part of the webinar or wish to listen to it again, a link to an archived version will be emailed to all registrants. With that, very many thanks for joining this OGT webinar today. Have a good day.